muscle contraction. So we need to know, what calcium is really doing when it comes to the sliding filament theory. So when there's not a lot of calcium, so it's saying a low intracellular calcium concentration, meaning there's not that much calcium inside the cell, this is when the tropomyosin is going to block the actin, the active or binding sites on actin. So the tropomyosin is going to be covering those little active sites. So if the tropomyosin is covering those, then the myosin cross bridges cannot actually attach. So in this case, we're going to have a relaxed state. That also means that the cell membrane is going to be polarized. When calcium comes out of the sarcoplasmic reticulum, meaning that there's going to be a higher intracellular calcium concentration, the troponin is going to basically change its conformity. And when it does that, it's going to pull off the tropomyosin. And when it pulls the tropomyosin off of those binding sites on actin, then we can have myosin heads attach. So at this point, when calcium is present and attached to the troponin, then we're going to have a one cycle of that contraction. Now the sequence of events for contraction, and we've gone through this before, you guys have seen this, so this isn't anything new for you, but the cross bridge formation, that's when myosin cross bridges are going to attach to actin. We're going to have that working stroke, which we call a power stroke, and that is going to pull actin towards the center, towards the M line. And then we need ATP to attach to myosin so we can detach the cross bridge. And then we have a breakdown of that ATP so that then that myosin cross bridge is ready to go again if there is another muscle contraction. So the next series of slides are just going to be pictures of what's going on, which you guys have seen this in the sliding filament theory little activity that you did online. So you can see that the myosin head is actually attached. It's going to go through its power stroke. We're going to then be able to detach it, so we need another ATP molecule. And then it is going to break down that ATP into ADP, so now it is ready for another contraction.